Today we have Sara Mostafavi coming all the way from statistics at UBC. We met in 2007 at the Max Planck Institute in Germany. Machine learning summer school. So if you like machine learning, you can go to machine learning summer school and talk to SVMs for 2 weeks. Google gives you free beer, so that helps. She just started at UBC in January. She's very strong in machine learning and applying it to important problems like cancer. Yes?
Kind of.
And she's also looking for students right now, which is relevant to many of you. She spent some, so U of T, with Clayton Morris, she did this cool thing called Gene Mania, which allows you to find relationships between genes. It's used by a lot of biologists. Spent some time in the AI lab at Stanford with Daphne Kohler, and went to Harvard, and now is at UBC. And we're lucky to have her at UBC and talking to us today.
Thanks, Mark. So I actually looked for a picture of MLSS, and I did find one, but I thought I should not share because there was a lot of drinking going on at night. Okay, so I'm going to tell you about inferring and analyzing gene functional networks, or actually graphs, for understanding biology from high-dimensional data, and I'll unpack all of that for you very shortly. But before I go on, I wanted to take 2 minutes to go over some basic terminology. And that's because of the frustration that I had myself with vagueness of terminology in biology. I think biology has a lot of interesting and challenging problems that computer scientists like you would be really interested to solve, but a lot of the barrier is the vagueness of terminology. And that, I think, has a good historical reason because of the history of the field. But for the purpose of this talk, I'm just going to take 2 minutes to go through this slowly so we're all on the same page. So all the cells in your body have a DNA, and DNA is the blueprints for all the genetic material. So genes are segments of the DNA that code for proteins. And for the purposes of this talk, I'm going to assume a one-to-one relationship between genes and proteins, although this is not necessarily true. So in order to make proteins, genes are first transcribed to this intermediate molecule called mRNAs, and mRNAs are translated into proteins. And then proteins come together and interact with each other, and that results in functional products, or that's how cells perform the functions that they want. So even though this DNA is fixed in all the cells in our bodies, the amount of genes that are produced or expressed could vary, and the pattern of interaction between genes or their protein products can vary depending on the context or the tissue and so on. So we have a fixed component and something that can vary, and this gives us a lot of flexibility. So what's happened in recent years in biology is that we've had amazing ability in, or amazing progress in our measurement technology. So we can now measure cells at various resolutions. So looking inside the cell, we can sequence the DNA and look at the differences in the DNA between different people. We can measure this intermediate molecule called mRNA, which is a proxy for activity of the gene in different contexts and different tissues. We can measure protein levels, and we can even measure interaction between different proteins. So by being able to measure all these things, we can try to answer very cool biological problems that we couldn't even tackle 10 years ago at all. So one data type that I'm going to talk a lot about is gene expression level. And this is one of the most prominent type of high-dimensional data that we can produce today. So this is measuring the mRNA, the level of mRNA level either in different tissues and different conditions. And we can do this genome-wide. That means we can measure all the genes at once. And typically, we have 20,000 to 30,000 genes. Okay, so what can we do with all this data that we're generating? We can try to answer basic biological questions. So what are the function of different genes? Which ones interact with each other? And how does that interaction result in biological process or disease? So that's basically, in the field, it's called completing the wiring diagram of the cell. That's how people usually refer to it. The second type of problem that we can solve is to understand different diseases better. So identify mutations and then identify consequences of those mutations, and based on that, identify therapeutic targets. So what are the challenges in doing this? Well, first of all, if any of you have worked with this kind of data, that you know the signal-to-noise ratio is very low. So we have lots of noise, sometimes very systematic noise that makes inference really hard. Second, we usually have very low statistical power. So we're measuring millions of variables, but only for a limited number of individuals or samples. So that makes overfitting, or that makes anything we do very prone to overfitting. And the third is scalability. So we need algorithms that could scale to the amount of data that we have. So in my lab, we use machine learning approaches that can integrate prior knowledge using structured regularization in a principled way to kind of tackle the first two challenges, and use well-motivated approximation to try to tackle the scalability issue. So in this talk, I'm going to tell you about two projects that try to address some of these challenges that I've brought up. The first project is the one that Mark mentioned briefly, is a project called GeneMania, which is an algorithm, an approach for combining multiple types of data in order to infer a function for uncharacterized genes. The second project that I'm going to tell you about is a hierarchical machine transfer learning approach for building tissue-specific regulatory network, and I'll tell you what all that means when we get there. So the first problem is about predicting the function of uncharacterized genes. The motivation behind this problem is pretty clear, actually. So according to really recent statistics, we know that about 50% of human genes that we know code for proteins have no assigned function. So we don't know anything about them. What do they do in the cell? And this is obviously a big problem for analyzing this data, because the end result of generating all these data, let's say we want to understand what's wrong in a given disease, we end up with a list of hit genes, but we don't know what those genes do. So we definitely need to address this problem somehow. So this observation has motivated a large number of algorithms that try to predict function of unknown genes based on what we know about genes with functions. So here's the basic idea. We've measured a bunch of different data types or data sets, and all of these measure something about the genes, even though they don't measure anything directly about gene function. So the idea is that we can use correlation and, for example, expression profiles between pairs of genes to learn something about co-functionality. The assumption being that if two genes have very similar expression across, let's say, a set of tissues, then those are likely to have similar function. And this is just basically the idea of using correlation between activity level or some properties of the gene to say something about their co-functionality. So this is the basic assumption, but obviously we can't just do this based on one data set and one function. But it turns out that if you think about all the different data sets that we have generated today, and there's hundreds of millions of data sets that we've generated, we can start to get a very good idea about the relationship between the genes themselves. So we not just have gene expression data, which is this data set here, but we've measured interaction profiles in some situation. We could have measured something about sequence composition of the genes. So we can try to build models that tell us about co-functionality between genes from all these different data sets. One term that I'm going to actually use a lot that I realized I didn't define is co-expression. And just this co-expression just means correlated expression profile. So in terms of what we're actually going to do, we're going to try to pose this problem as a binary classification task. So here's how we're going to deal with it. We're going to assume that for training, we have a set of labeled genes that we know have a particular function of interest. So for example, I'm showing a few genes here. We know that these genes are involved in regulation of cell cycles. As our training, we would like to use all sorts of available data in order to make some prediction about which other genes we think have the same function. So our training genes, I'm going to sometimes refer to them as positive genes or query genes. And obviously, if you think about this problem, we're not going to use, we want to potentially be able to use all sorts of available data, but for any particular prediction task, we might only want to use a subset of that. And ideally, we want to have an automatic way for selecting which of these data sets we should be using in a particular prediction task. So the requirement that we have for this problem are scalability. We have lots of data and 10,000 to 20,000 genes. We have lots of functions that we want to potentially make predictions for. And also, we want to be able to do feature selection in a realistic way. And at the end of the day, it would be ideal if we can have this algorithm implemented online so biologists can actually use this on all of the available data in the world, hopefully. And this was what we set out to do. So the algorithm that we came up with actually broke down this problem into 3 steps. And it turns out that these 3 steps are essential for having a fast algorithm that we can implement online. So the 3 steps are as follows. In the first step, we're going to actually download all the data that we can from public repositories and convert all of them to a common representation. And for that, we're going to use graphs. And I'll tell you how we do that in the next step. So each node in the graphs each node is going to be a gene, and the connections are going to reflect some measure of correlation between the genes. In the second step, which is going to be online, we're going to find a weighted combination of these underlying graph that is best suited for a particular query. And in the third step, we're going to use the combined network or a combined graph that was particularly constructed for a given query to make prediction about which other genes are likely to be positive. So we'll go through each of those steps. So in the first step, you want to take all sorts of available data and represent them as graphs. How do we do this? So we have basically 2 types of data. One data is actually already represented as a graph. So these are direct interaction-based data where we actually measured either physical interaction or some sort of interaction between proteins, product of genes in the cell. So those are already given to us. But then the second type of data that we could have is a profiling-based data, where for each gene in the genome, we measured some sort of profile. It could be activity of the gene. It could be in terms of mRNA level. It could be its protein level. It could be sequence composition and things like that. So those are profiling data. We're going to take those and translate those into a graph where nodes are the genes and the edges are the similarity based on correlation coefficients. And we're going to adopt that to be suitable for depending on the data type. So each of these graphs now represent a separate data set. And we're going to encode those as weighted adjacency matrices. So now that we have all of this, and we ended up actually generating, I think, about 2,000 different graphs from 2,000 different data sets that we downloaded, what we would want to do is have an algorithm for automatically combining these given a query. But before I tell you about that, I want to just tell you about how we would make predictions given just one graph. So here, let's go through some terminology. We have our one graph. The nodes are the genes again. The connections are some weighted combination, some weighted evidence for co-functionality between the genes. And we're going to have 2 input variables. So our graphs are represented as the matrix W, and the non-zero elements refer to the edges. Then we have this label vector Y, which is +1, 0, or -1, and Y is the length of the number of genes that we have. So in some examples, we're going to have some positive genes. These are labeled as +1. Most genes in the genome we don't know anything about, and these are going to be labeled as 0. And we have some negative examples, not always, but we could have some negative example in some situations. So the idea is that given this graph and a set of positive and negative example, we would want to assign a score to all the other nodes in the graph that reflects how likely are they to be also positive or predicted to have the same function. And we're going to do that by assigning or computing a score for each gene, which is encoded in this vector f, that entry fi is a score of node I that tells us about how likely is that gene I to have the same function as our query genes. So we're going to do this by using this very famous algorithm called label propagation. And this algorithm has been reinvented in many different fields and has many different names, and many different formulations, actually. But I really like this formulation, so I'm going to go through this. So the idea is that we want to infer these FIs, which is the scores for the nodes. I think there's a question.
Yeah. So I can see like one node can have multiple edges coming into it as well.
Yes.
My question is, the nodes from which those edges are coming.
Sorry, can you repeat that?
The source nodes for those, say for example the topmost node here,
Yeah.
can the source node for those two edges be labeled differently? Can that be a case, or like, I mean, so essentially age is representing the likelihood of two nodes being similar, right?
Each edge is some measure of, it's a weighted evidence for co-functionality between the connected nodes.
Okay.
So let's go through this and maybe that will clarify things. So the objective that we're going to use is composed of two terms. So the idea is that we want to estimate a score for each node in this graph, right? And that's going to be a continuous score. In order to do that, we're going to optimize this objective, where the first term is that we want the score for genes that we know their label, either +1 or -1, to be similar to that original label. So that's just doing well on our training data. The second term is a regularization term, which just says genes that are close together, so they're directly connected together based on this encoded matrix W, should have similar scores. So we're penalizing ourselves if the score assigned to node I is very different from score assigned to node J, and those two are connected together strongly. So the reason why I really like this algorithm and we ended up using it is that it turns out that it has a closed form solution. So you could actually write it in matrix form, optimize it, and you end up with the following system of linear equation. And all you have to do in order to compute the score for each node is to solve that. And that happens to have actually a very fast numerical solution, especially if you have sparse graphs, which we do in this case. So I'll come back to this later, but the other thing that I wanted to mention is that now that we have this formulation, what we can do is that modify this objective function, not just to use one graph, which was the example we went through, but multiple graphs. So the idea is that we add this term in the middle that says we want to find a weighted combination of the graphs or the networks that gives us best performance on the labeled example. And of course, we can't do that without adding some regularization. So the alphas are the weights assigned to each of our graphs or each of our networks. Does anybody have any questions? Does that answer your question? Or okay. Okay, so we ended up applying this algorithm on a variety of...
Yeah, I have a question. So back when you just have one graph, it's just neighbors. So the sort of flow of similarity is really just over a single edge.
No, because what happens is that you don't know these FIs, right? So it happens that it gets propagated, and you'll see a formulation by that. It gets propagated through the network, but so it ends up being, a score ends up being related to all your neighbor weighted by their distance to you.
Okay.
How does this formulation relate to the one on the next slide? I didn't catch.
Oh yeah. Okay. So. So from the scalar version to the matrix version, is that clear?
Okay.
So then you just take that, and now you add a summation for graph Laplacian for. So for each graph, sorry, I kind of skipped this because it's a lot of detail, but so for each graph that's represented as W, you have a Laplacian, which is a diagonal matrix encoding the degree of the graph minus the W. That's your graph Laplacian L. So you have a weighted combination of those, and you have an additional regularization based
the
on.
alphas have to sum to 1 or something? Is this a probability or something?
Yeah, you have to have some constraint on the alphas, and the easiest version would be that they sum to 1. So, okay. So one thing that I wanted to mention is that that actually turns out to be kind of computationally intensive. So we ended up approximating that using a very simple two-step algorithm that resulted in almost the same performance on a lot of our benchmark. Okay, so the easiest way to compare your algorithm to other approaches is to enter a competition and hope that everything works out for you. Because you get graphs like this. You just enter your results, and then you get compared to all other algorithms that are trying to do the same thing. And that's what we did. And so this competition called MouseFunc, which put aside some test data that didn't show anybody, and this was basically a functional annotation for genes where their function wasn't previously known. They gave us some training data that we trained on and made prediction for the test genes. And then they gave us the performance, and it was really nice because it turned out that so each set of bar is a different evaluation criteria that they use. And it turned out that the approach that we developed, which we call G-Mania, performed as one of the best, not all the times, but in a lot of the cases. And especially when it performed the best was the cases that we really cared about, which is the cases where you only have between 3 to 10 positive examples, and those are the exact ones that usually biologists want to solve. Okay, so what I wanted before I move on to the next topic, I wanted to come back to this formulation. So I've changed it around a little bit, but for the reason that you'll see in a second. But I told you that it turns out that label propagation has a simple closed form solution, and the solution is just to solve a linear system of equations that's on the left here. So when we were thinking about this problem and how to make it faster, we realized that you could actually write that the solution to, well, you can write a matrix inverse given certain properties are satisfied, which were in our case, using an infinite series. So terms in this series now make a lot of sense and allow us to interpret what this algorithm is doing in a very intuitive way. So let's go through the terms. So each term is basically w raised to a power, increasing power, multiplied by this vector y, which was our label vector. So what's the first term? So here's a very simple representation. This w is encoding the weighted adjacency matrix. This is our graph here, so it's equivalent to this one. Our label vector y, we have one example, positive example. If we multiply w by 1, we end up with a vector here, which basically has elements proportional in value to their weighted direct connection with the positive node. So in this case, you can think of it as we're starting with this graph, and we're propagating the positiveness to its direct neighbors, let's say. And the second step, which is the second term in that algorithm, what we're doing is that we're propagating the positiveness to neighbors that are 2 steps away or have a path of length to the positive node. And you can see a cartoon of that here, and that's what that score vector over there is what we're ending up with. So if we go through this summation, we basically end up with one vector for each of those terms, and these reflect the amount of positiveness that a node has received based on its neighbor at certain distances. So 2 important observations here. One is that we can actually truncate this series pretty early, because it turns out that w raised to the power of n on real-world network actually converges to stationary distribution of that matrix really fast. I won't go through the details of that, but that's actually a pretty simple fact. So when we're applying this to real-world data, we can actually truncate that series, maybe 2 or 3 after 2 or 3 steps, and you would end up getting identical results. The second observation was that this lambda, which I kind of didn't talk about, so if you see a lambda, it should be between 0 and 1, and that's the one parameter that the model has. What is that lambda doing is it's weighing these different steps. And because it's between 0 and 1, we're enforcing a geometrically decreasing weighing scheme. What does that mean? That means your direct neighbor always have more influence than you, and exponentially more than your second degree neighbors, and second degree neighbors have more influence on you than your third degree neighbors, and so on. So this is very intuitive, but that's not the only weighing scheme that we could have. And actually, it turns out that if you read the literature on these kind of problems, there's two types of labeling problems that people have identified. So there is one type of labeling problem that's called assortative mixing, and this is what exactly label propagation is optimized for, where direct neighborhood in the graph actually tell you about co-labeling. So in this example, we have a bunch of nodes here, and with the darker red circle, I'm depicting the nodes that have the same label. So everything that has the same label is connected to each other. And they're not really connected to other genes over there, or nodes over there that we think are not sharing the same labels with them. We could have a different type of scenario, which is called disassortative mixing, whereby direct relationships between the nodes doesn't tell us anything about co-labeling. So in this case, we have a set of nodes in the outer part of the graph and a set of nodes in the inner part. And the ones on the outer part are directly connected to the inner nodes, and they're not sharing any labels with each other. So in this case, this really breaks the assumptions of label propagation, whereby direct neighbors have the most influence over your predicted score at the end.
What do the labels mean here?
So here it could be that they're positive. So in our example, it was they have the same function. So each node is a gene, and the label is, are those genes share the same function or not? Yeah.
When you say function, you mean like overall, like big time function?
So I mean, I'm being a little bit vague about that because we want to leave this up to the person that's querying our algorithm. So all you have to do is put in some genes that you think are related to each other for some reason, and you want to identify other genes that should be related to your input genes. So the example that I gave in the beginning was genes that are involved in regulation of cell cycle. So if you have 5 or 6 genes and you know these are involved in regulation of cell cycle, these are your positive genes, and you want to find more genes like them.
So is an example of disassortative mixing when there's like, say, 2 inhibitors on the same gene, and so you'll see one or the other, but not both? Is that a
Yeah, that could be an example. And there's a lot of example in genetic interaction data, which I won't go into details, but maybe you know about what that data represents. And it's a very similar situation. So, so recognizing that you could have both of these scenarios, and ahead of time you don't know which one you might have, we came up with very simple algorithms. So our idea was that first we're going to use the truncation observation and only look at the first 3 terms in that Taylor expansion. And this was because of empirical reason why we stuck with 3. So to implement this idea of automatically determining what weighing scheme to use, we're going to have 3 free parameters. So lambda 1, lambda 2, lambda 3, that are going to weigh those 3 terms in the Taylor expansion. And the way we're going to determine these is based on a simple grid search to best fit our training data. So in this example, let's go through it. We have our graph here on the left, and our labeled nodes, which are our example of positive nodes, are the darker or the bigger circle. We have 4 examples of training data, the 4 positive examples. Each term in the Taylor series is shown there, and the size of the node is proportional to the score they've received from the positive node. And then we're going to assign a weight to each of these subnet or networks or graphs that results in a weighted combination of them that best recapitulates that training data. And to do this, what we would have to do is assign a negative weight to the first term. And if we do that, then everything in the inside will have a lower score than everything in the outside. And in fact, if we do that, we can learn these weights with our algorithm 3-prop, and this would be our prediction. So all the nodes in the outer circle now have a higher score than the nodes in the inner circle. But if you apply label propagation, you would get the opposite of that, and that's because label propagation is making the opposite assumption compared to the actual observed labeling pattern on this graph.
So is it still a quadratic objective function?
In terms of cost,
So is you're defining how good f is
Yeah.
somehow? Is that still a
oh yeah,
quadratic
yeah.
function?
So
You're not really learning anti-correlations. You're just learning correlations at different steps away.
yes, you're learning how to weight those
Yeah,
correlations.
but you're not saying that I'm supposed to be different.
No,
than my
because
neighbor.
it's just based on distance. Like if you're 1 or 2 steps away, you always will have either be positively weighted or negatively weighted.
You only have positive examples, is that right? So
Yeah.
your regularization is sort of stopping you making everything positive. So how do you, when your data, how do you measure, how you evaluate it if you only have positive examples?
Based on your goodness of prediction of those positive example, and you're paying some penalty for, if you had a 0 label, I guess you are,
But he's saying you're only getting +1 labels.
Yeah.
here. So where does the 0, where do the -1s come from?
oh, so the -1s is you could be supplied with a negative example, or if you're doing this offline, you could assume that genes that were assigned a function but not the one you're interested in are negative examples. So you would have to make some biological assumption about what is negative. Wait, did I answer your question? So we applied this algorithm on very different tasks, and I'm showing you some example here. So predicting gene function from different types of networks. So genetic networks are actually networks that turn out to have this assortative mixing property. Protein interaction networks are networks that turn out to have assortative mixing properties. And then we also looked at different, not biological tasks, but different tasks, for example, predicting political view and from political blogs, co-citation based on the blogs, and predicting gender from friendship networks from Facebook. And it turned out that in all of these cases, our algorithm, even though it was only using the first 3 terms in that Taylor expansion, was performing better than using label propagation. And that's because we actually have these 3 free parameters that could allow us to determine what is the weighting scheme that should be used. Okay, so in the second part of the talk, I'm going to switch gears a little bit and tell you about inferring these graphs from gene expression data, and in particular, the task of inferring multiple different graphs multiple graphs that represent the connectivity between genes in different tissues. Unless you guys have questions about the first part. Yeah?
So do you think the same approach can be used to say for isoform function instead of just gene function?
Yeah, you could do, just your graphs would be bigger because you have multiple isoforms, but per gene, but the same thing could be applied.
What is the scale of the data? I mean, what is the size of the W matrix? It's like the number of graphs times the number of nodes, right?
The scale of W matrix, or you mean what's the dimension of the matrix W? It's n by n if n is the number of genes.
It does not depend on the number of graphs you're trying to merge.
The number of data sets. So we have different W's that correspond to different data sets. So if you have k data sets, we're going to have k W's. Each one is n by n.
And in this, each node has like a scalar attribute, right?
Yes.
Suppose you have vector attributes at each node. Do all these tricks still carry on?
So each node does have an attribute, so that's a +1, 0, or -1 label. Yes. So you have one label vector. They're not different for the different networks. You're assuming that that's fixed for a given prediction task.
What if different genes repeated in different data sets?
So you have different connections between the genes, right? If they're repeated in different data sets, is that what you mean? So they're going to have different connections depending on which data set you're talking about. And the point is that by aggregating those connections, we can make better predictions.
Yeah, do you consider, you're considering that by aggregation? Like
So the way we aggregate it is by coming up with this weighted combination of the graphs and not just using one graph that's determined by the prediction task. Okay, so in the first part of the talk, I told you about the problem of using graphs that represent functional relationship between genes to make predictions about gene function. In the second part of the talk, I'm going to actually tell you about how we infer these networks from very high-dimensional data. I'll tell you about the motivation in a second, but why we want to do that multiple times. But just to motivate this problem a little bit more, so you saw that these graphs are very fundamental. If we have accurate representation of functional relationship between genes, we can infer something about unknown genes or uncharacterized genes. But these graphs are also fundamental because we need them to analyze, to understand different diseases and how mutations in different diseases result in processes that are abnormal, and also to identify effective target for therapies. So having a good understanding of what these graphs look like, what's the relationship between genes, is very fundamental. So the motivation for learning different graphs for different tissues as follows. As you know, all of our cells and tissues have the same DNA. But it turns out that, you all know this, they have very different functional and physical forms. So you can look at a brain cell and a heart cell. They have very different functional and physical properties, even though they were both based on the same DNA. And it's widely believed, and there's evidence for this, that some of these differences, some of these tissue specificity is underlied by differences in so-called regulatory networks, so the interactions between genes or product of those genes and different tissues that are different. So even though you're starting from the same set of genes and those result in the same set of proteins, the activity level of those, so expression of those, and the way they interact with each other could drastically be different in different tissues, and that results in tissue specificity. So up until very recently, most of the data that was available to build these networks or graphs from was gene expression data, and you can widely measure gene expression data only in blood. So there wasn't a lot of additional data from different tissues, and humans specifically, to understand these differences at a global level. But very recently, a really big consortium called the GTEx generated a lot of relevant data sets. So in particular, GTEx generated gene expression data for over 30 different human tissues. And here I'm showing you the number of samples that's available for those different tissues. And the reason that I'm showing you this is to highlight that some of the tissues we have a lot of samples, or reasonably a large number of samples, and some of the tissues we have a much smaller number of samples. And this will become important later. But here's what that data actually looks like. For each tissue, we have gene expression level for a set of individuals. So the number of genes is always fixed. So we have about 20,000 genes that we're always measuring. And we're measuring the expression level of those genes and the number of individuals for different tissues. And the number of individuals is what's on the bar graph. So we have multiple of these matrices. So now the goal is to take each of these matrices and convert it into a graph or network. And from now on, I'm going to refer to these graphs as regulatory network because they're based on gene expression data. And when we do that, the assumption is that co-expression tells us about co-regulation. That's why they're called regulatory networks. So we want to take each of those expression data and build a regulatory network. And then we want to be able to compare those to understand the principles of tissue specificity better. But doing that, we're faced with a lot of challenges. The first is that for each tissue, we have at least 10,000 genes, and this is an underestimate. So we need to be able to estimate millions of parameters. And sometimes we don't have enough data. We have 10 to 20 samples, and that's just small number of samples in contrast to the number of variables that we have and the number of parameters that we have to estimate. So we need to estimate millions of parameters here. The varying number of samples across different tissues is a problem because we have different statistical power in different tissues. So for some tissues, we're well powered, for other tissues, we're not. And if we're not careful, we can't really compare the networks that we're estimating for different tissues. So the goal here is to estimate these network in a way that is accurate and robust and comparable, because at the end of the day, we're really interested in this problem of tissue specificity. So in order to address this problem, what we end up developing was an approach that's based on hierarchical transfer learning built on top of Gaussian random fields. And I'll tell you all of that. But to tell you that, first I'm going to review two very simple quick concepts. So the first is that if we have three random variables, x1, x2, and x3, and we have some true relationship between them, depicted in the graphical model here. So if you know there's a causal chain between them, then x3 is independent of x1 given x2, and we can just read that off according to that graphical model. So if we had some realization from these three variables, and we just measure the correlation between the variables based on the realization, we would end up reconstructing a very dense graph between these three, because everything is correlated with everything, and correlation just tells us both about direct and indirect relationships. But if we measured conditional correlation or partial correlation, we would end up recovering this sparser set of sparse set of relationship, and that's what we basically want to focus on. We don't want to capture all the indirect relationships. So we really need to compute these conditional correlations and not just correlations. So a really cool fact is that if you assume that the three variables, x1, x2, x3, are jointly Gaussian, then you can compute the covariance matrix between them, and that tells you about direct correlations, or that gives you actually the correlation coefficient between them by normalizing the off-diagonal elements in the covariance matrix. And that gives you the denser set of relationship. But the cool thing is that if you actually took the inverse of that covariance matrix, you looked at the precision matrix, then the off-diagonal elements actually tell you about the conditional or partial correlation between pairs of variables. So now you've estimated a sparser set of relationship that you could use. So Gaussian Markov random fields are models that assume that a set of variables are distributed according to a multivariate Gaussian, and the goal is to estimate the precision matrix that tells us about the relationship between those set of variables. So in our case, where we have, for a given tissue, we're going to have a matrix X, where we have the genes, and the genes are the variables, and we have a set of samples. We can assume that each sample, the likelihood of each sample under our model is given by a Gaussian distribution. Oh, you can see this. OK. So each xi is the gene expression profile for each sample, and we assume that's coming from a Gaussian distribution with mean 0 and a covariance matrix. And in Gaussian random fields, we want to basically estimate this covariance matrix that best fits the data. And if you take the inverse of that, that gives us a sparser set of conditional correlations between the genes. So, yeah, we can actually optimize this objective function. And it turns out that the solution is, again, closed form and very simple. So not surprisingly, the precision matrix that we're looking for is given by the inverse of the sample covariance matrix. But we can only do this if we have full rank matrices. And in our case, the inverse is going to be highly unstable. So we can't actually invert the matrices as written in that objective, and we need to add some regularization to our objective. So the approach that we take adds this regularization term here, which is the L1 regularization, enforcing kind of sparsity or encouraging sparsity in our precision matrix. So it turns out that this objective still has a reasonably fast solution, and we can solve this for one tissue at a time. But what we really want to do is estimate these for all of our tissue in a way that's accurate and kind of realizes the differences in statistical power that we have between the different tissues. So in order to do this, we're gonna adapt transfer learning to our setting. And here's how it works. So the principle of transfer learning is that if you have two related learning tasks, then you can share data or parameters between those two tasks. And empirically, this improves your estimation of the parameter or your performance on test set. So in our case, in our context, we have a regulatory network for heart and a regulatory network for blood, and each of those are going to be represented by non-zero, or each of those are going to be captured by the precision matrices that we're estimating. The assumption is that we're going to have a lot of shared edges between the two tissues because there's going to be a lot of biological processes that are conserved between tissues. So what we would do in practice is we have our precision matrix for heart and precision matrix for blood, and we could encourage similarity between them by introducing another regularization term. For example, L2 regularization, and that's actually what we end up using. But our case is more complex because we don't have just two tissues, we have a lot of different tissues, and there could be different level of similarity between tissues. So for example, heart and muscle are going to be more similar to each other compared to heart and blood because heart and muscle share more biological processes. So how can we kind of encode this in a systematic way? The way we came up with was, the idea was to use a hierarchy that defines the relationship between tissues. So we're going to assume that somebody gave us a hierarchy that tells us about the relationship between different tissues, and it's actually pretty simple to derive or get this hierarchy based on prior knowledge. So here I'm showing you the relationship between the 30 tissues or so that we have in our data. And you can see you see expected relationships. So we have a bunch of brain tissues that are clustered together at the top, and then we have heart and muscle somewhere clustered together in the middle. So the idea is that, the intuition is that the closer the two tissues are according to this hierarchy, the more we're going to enforce that regularization, the more the precision matrices should be similar to each other. So the way we actually implement this is as follows. So we have, in this example, we have 4 different tissues. For each of the tissues are a leaf node in that hierarchy. We're going to estimate a precision matrix for each leaf node and each internal node in the hierarchy. And if we do that, then we can basically say that we want our each precision matrix for each node to be close to its parent. And if you do that, it turns out that you have this property that tissues have similar precision matrices depending on their relationship on that hierarchy. So I won't go through the formulation, but that's just the basic assumption behind it. Yeah.
Is there any specific reason you use the 2 norm for similarity between the matrices?
Can you say it again?
Is there any specific reason why you use the 2 norm?
The 2 norm? Oh yeah, for
comparing
scalability.
databases.
Yeah, precisely for scalability. So the nice thing about this formulation is that, so we have L1 norm for each observed tissue, which is each leaf node, enforcing sparsity. And then we have L2 norm relating each tissue to its parent tissue. And by splitting the objective, by writing the objective in this way, it turns out that we have an iterative algorithm where we have 2 types of step. First step is that we're estimating precision matrices for all the leaf nodes, and we can do that independently for each leaf node. And the second type of step, we are computing the precision matrices for the internal nodes. And that has a closed form solution. So this really speeds things up, and that's by writing the objective in that way and making those choices, we were actually able to apply this to 35 different tissues and about 10,000 genes all at once.
Do the internal nodes show up as part of the likelihood?
Yeah.
In the prior?
They're part of the likelihood. So you see this is the summation from k to 2k plus 1. Yeah. So each node has, oh, do you mean, are they here too?
The left one is the likelihood.
Yeah. The right one's regularization, yeah. They are part of here too.
Okay, so how do you make the sigma k for an internal node?
Sigma k. How do you?
How do you get the sample covariance for?
Oh wait, do I?
No, it looks like you're.
No, we don't have. Yeah, we don't have. We only have the.
Okay, and the other issue is these are covariance matrices, so they need to be positive definite. So
Yes.
how the thing on the right could cause you trouble. Because your solution might not, there might actually be no solution to this problem.
Yes, potentially.
Okay.
Wait, the thing on the right?
So the thing on the left will make sure that your S1 to K are
Yeah.
positive definite.
Yes,
The thing on the right,
the right.
that's the only where the internal nodes show up. There's nothing, there's no log determinant there to keep
Oh,
it
yeah.
inside the positive definite cone.
We might have a log. I have to check that out. So, yeah, let me get back to you on that. But yeah, there is definitely, we do enforce that they are positive definite in the internal node. I don't remember how.
Got a related question, I think, but I'm not sure. Do you actually, there is a data set for the internal nodes, which is the union of tissue A and tissue B. Do you use that data set ever?
Union of tissue A and tissue B. So in terms of the internal node, it's going to be the average of the networks for
There's a
tissue A.
data
And,
set for it, right? There's all of the
oh
graphs,
yeah, I see what you
right?
mean.
Another data
No,
set.
not in that way directly,
Okay.
but it relies on but it's similar to the network for tissue A and B, but not using it directly to estimate it. But yeah, I see your point. It's a good, yeah. Actually, that might be what we do, but I have to double check.
That was related to my
Yeah.
first question. Is
Yeah, yeah. Okay, so how are we going to actually evaluate the objective? The most basic thing we can compare it to is a baseline where you are independently estimating a precision matrix for each tissue. And the way we do this, I'm just showing you three examples, but so you have this lambda parent, which enforces the similarity between each leaf node and each of its parents. So if that is 0, there is no parent node, and each tissue is estimated independently. So you can see that the likelihood, and I'll tell you what actually we're measuring. So what we're doing is that we are doing a cross-validation where we're leaving out some of the samples as test samples. We're computing our precision matrices on the training data, and then we're saying what's the likelihood of the test data under our model. And we can compare that to for our approach and for precision matrices that are learned for each tissue independently. And in all cases, we saw an improvement for the likelihood of the test data under the models where we shared, where we had this kind of hierarchical regularization. On a biological side, we looked at two quick things that I'll mention. One is that, so we estimated these precision matrices that encode the regulatory networks. We know how close the tissues are in the prior space where we observed that hierarchy over the tissues, but is that consistent with what we estimated in terms of our precision matrices? So to evaluate this question, so here I'm showing you the distance between tissues based on prior knowledge, and this is the number of edges that's shared between the tissues based on our precision matrices. And then what you can see is that the relationships that we're estimating are pretty consistent with what we started with. And this is to some extent expected, but it could have been that if our prior knowledge is totally not consistent with the data, then we would see some deviation from this pattern, but that wasn't true. The second thing that we looked at was more of a biological question. So we were really interested to see what types of genes share their neighbors a lot, depending on what tissue we look at. So the way we asked this question was to look at two types of tissue, two groupings of the tissue, and asking what are the hub genes that are have many connections in one set of tissues, but no set of connection in a second set of tissues. And when we identify a handful of these genes that have very different number of connections depending on subset of tissues, and it turns out that most of these genes are very important known disease genes. So I'm showing you one example of that here, where we found one gene that had a reasonable number of connections in non-brain tissues and a lot more connections in all the brain tissues. And this gene is Huntington gene, which is known to cause Huntington disease. So that's one thing that we're following up on. So in conclusion, I told you about 2 separate projects for using a large number of genomics data to better understand basic biology. And the first part of the talk, I told you about an approach for combining multiple different data types by representing them as graphs, and it turns out that that combination, being able to use multiple data types, really gave us an accuracy that we couldn't have if we just looked at one data type at a time. In the second part of the talk, I told you about an approach for building regulatory network for different tissues in a way where we couple learning based on the closeness of different tissues. And this gave us better performance in terms of identifying biologically interesting genes and statistical performance on test data. So just to acknowledge people that were involved in this work. So the first part of the work was Dom and I was at University of Toronto working with my advisor, Quaid Morris, and also Anna Goldenberg. And the second part of the talk, I was at Stanford, and a lot of it is work by a great student, Emma Pearson, and a colleague of mine, Alexa Spattle, and Daphne Kohler. Thanks so much for listening.
One question for Sarah?
Yeah.
So are the methods robust to say noise in your microarray measurements or whatever the initial gene is?
So the first part of the talk, you mean, or the second, or both?
The first part.
So the idea is that none of the individual data sets are going to be that robust, and there's lots of noise. But when you have a lot of different data sets and you're combining those, and there's different kind of systematic biases in different data sets, by averaging them, hopefully you're getting rid of the noise that's very specific and technical to one data set or data type.
In the second half, there's a reason, for example, in a phylogenetic tree that it's a tree because of evolution. Is there any reason why this should be a tree as opposed to just sort of trying to work out the similarity between the node?
Well, it makes intuitively sense in biological setting because all the tissue, there are different levels of similarity between the tissues, and all the tissues come from the same individual cell. So you can see that there is a phylogenetic relationship between them. But I don't know how well it would work. I don't know how much that's contributing to our accuracy. It could be that maybe you just want to have, you don't need a phylogenetic tree.
And I was saying, why do you assume a tree?
And why not? Just because it made biological sense, I guess that's the only reason.
So it does make biological sense for the tissues.
Yeah.
Okay, let's thank Sarah again.